Welcome to the Right Science Daily Revision Boost. The formula for today is that for speed. So speed in meters per second is the distance in meters divided by the time in seconds. What we're going to look at in today's little biology blast is DNA. Hopefully we remember that in our eukaryotic cells, which are plant and animal cells, the genetic material is DNA, which is found inside the nucleus. Inside the nucleus, it's organised into structures called chromosomes, and a section of the DNA on the chromosome is referred to as a gene, and each gene codes for a protein. When we talk about DNA, we need to remember that everyone has a unique DNA code, with the exception of identical twins. When we're referring to a gene, we refer to a short section of DNA that codes for a certain characteristic because they code for a protein. And we need to remember that not all genes in a cell are switched on all the time. So what we find is some genes are switched off in a certain cell in order to stop them making a particular protein. So in a red blood cell, you'll have certain genes switched on, but in a white blood cell, it'll be different genes. DNA is an example of a biological polymer. It's made up of two strands which are bonded together using bonds between the bases to form this structure called a double helix. The strands are made of the monomers, so the individual units called nucleotides, and the nucleotide is made up of the sugar, deoxyribose, a phosphate group, and a base, either adenine, thymine, cytosine or guanine, and the structure for it is shown in the bottom right. When the monomers of those nucleotides make the polymer, what happens is the nucleotides join together to make a sugar phosphate backbone, and then the bases, so the adenine, the thymine, the cytosine and the guanine, bond via complementary base pairing, A's with T's and C's with G's, to connect the two strands of DNA. If you're not doing the higher tier, you can shut your brain down now, and if you're only doing combined science, again, you can shut your brain down now for a moment. Those of you doing higher tier biology, we need to know about the process of transcription. So this is where we use the DNA to make proteins in a process called protein synthesis. And the first step of this is transcription. So what happens here, the DNA unzips so that we expose one strand of our DNA there. The free RNA bases will then pair with their complementary base pairs to make a single-stranded mRNA molecule, remembering that when we're talking about RNA, it doesn't have thymine. So wherever you see adenine in the DNA, then the RNA will have a U for uracil. Once we've formed the mRNA strand for that gene, it detaches from the DNA and leaves the nucleus, where it will then travel to a ribosome. So transcription, we write the code into the mRNA. The second part of protein synthesis is translation. So this is where the mRNA attaches to the ribosome in the cytoplasm. Then the mRNA bases are read in triplets, so sequences of 3. What we find there is each triplet codes for an amino acid, and each amino acid is joined using complementary base pairs, as you can see in the diagram, and the amino acids join together via the bonds to make a polymer chain. And the sequence of the amino acids determines how the protein folds, and this using the mRNA to join the amino acids is translation. Obviously, don't forget that today's daily formula is that for speed, which is measured in meters per second and is distance in meters divided by the time in seconds. Today's chemistry check is about separating techniques. So we have a look at filtration first of all, which is where we're separating an insoluble solid from a liquid. So the filter paper's got these tiny little microscopic holes in it, and they're small enough to allow small molecules through, but not the larger ones. So the sand gets left in the top, the water can go through. So the sand that stays in the filter paper is called the residue, and the water that passes through is known as the filtrate. Our second process is crystallisation, which is used to separate a dissolved substance from a solution. So what we need to do here is set up the equipment as on the right there, and we need to heat the solution gently until it becomes saturated. When we're talking about a saturated solution, that's one where no more solute can dissolve at that temperature. So when that happens, crystals will form, and then as the solution cools, the solubility decreases, so more crystals form. Once we've actually got those crystals, we can use filtration to separate them from the remaining solution and then dry them in a warm oven or pat them with some filter paper. Our third process is simple distillation, and the setup is on the right there. So we use simple distillation to separate a solvent from a solution, and the key thing here is that the solvent must have a lower boiling point than the solute, so that when we heat it, the solvent evaporates, and then as it passes down the condenser, it gets cooled and condenses, and then we can collect that on the far side in our test tube. The fourth separating technique is fractional distillation. So this is used to separate two or more substances from a mixture in the liquid state. So something like ethanol and water. Key thing about this is that each substance must have a different boiling point, and we use a bit of equipment called a fractionating column. So as we heat it up, the one with the lowest boiling point will turn to a gas first of all. As it passes down the condenser, it condenses to form the solution which drips out at the end. Then, as we go on to the next boiling point, we can collect the next fraction. The last separating technique is chromatography. The first version of this is thin layer chromatography, which has a stationary phase of a thin layer of silica or alumina powder spread over a plate of plastic or glass. The mobile phase is the solvent that moves up the plate. So we put the solvent into the tank to about 1 centimeter depth, mark the baseline in pencil, put a small spot of the sample on the baseline, put the plate into the tank, and then we take the plate out before the solvent reaches the top. We can then be asked to calculate the RF value, which is the distance travelled by the substance divided by the distance travelled by the solvent. The second type of chromatography is gas chromatography. So we turn the sample into a gas when we inject it into the column. We use a carrier gas to push it through, which is an unreactive gas. Then different components will take different times to travel through the stationary phase, which is the particles in that little coil there. What we find is that then is picked up by the detector as it gets to the other side, and that detector sends a signal to the computer as each component leaves the column, and we get a chromatogram on our computer screen. And just in case you've forgotten, today's daily formula is speed in meters per second is the distance in meters divided by the time in seconds. In our physics prep today, we're going to have a look at Newton's laws. So Newton's first law of motion states an object will continue to stay at rest or move with uniform velocity unless a force acts on it. So in order to change the speed or direction of an object, we need a resultant force. If that resultant force is zero, speed or direction doesn't change. So make sure that you do remember that if an object has a steady speed, then that means that the forces are balanced or equal. Newton's second law states that the acceleration the resultant force produces on an object depends on the size of the resultant force and the mass of the object. So if the resultant force is not zero, then it can change the speed of the object, it can change the direction of motion of an object, or it could change both direction and speed of the object. So basically, if this isn't zero, it will accelerate the object. Newton's third law states for every action there is an equal and opposite reaction. So this is talking about the pairs of forces that arise when objects interact. So in any interaction pair, each force acts on a different object. The forces are the same size and type, and the forces will act in opposite directions. When we're talking about forces, we need to remember there are two key types. Non-contact forces are forces produced because an object is in a field, so they don't need to be in contact for the force to act. A field is a region where an electric charge, a magnetic material or a mass experiences a force, and are three examples of the non-contact forces: electrostatics, magnetism and gravity. The second type are the contact forces, and these are forces that only act when an object is in contact. And what we find here is that when we're drawing our force arrows, then they're drawn from the point of contact. So this will be things like friction. And just in case you've forgotten, today's daily formula is speed in meters per second is the distance in meters divided by the time in seconds. Now we've had a quick recap, then head on over to socrative.com and test your knowledge on the topics from today. The room name you need to enter is Right 2662. Good luck! If you want to know more about the topics covered in today's little daily booster, then head on over to the B1 playlist for DNA, the C2 playlist for separating techniques, and the P2 playlist for Newton's laws. Hopefully you'll be back again tomorrow for our next little daily booster.